about the maintenance of nucleus seeds and breeder seeds in seed technology course of 4 semesters. Today we will discuss all about this maintenance of nucleus and breeder seeds of different crops. So we know very well that generation system of seed multiplication, very known that nucleus seeds is multiplied to that of the breeder seeds and then foundation seeds, then certified seeds. The ultimate result is the certified seeds which are used by the farmers. So the genetic purity is essentially maintained in certified seeds. So for this we must maintain the nucleus and breeder seeds true to the type and genetically 100% pure. Because if 0.1% impurity is present in the nucleus seeds as well as the breeder seeds, it can multiply by 20% in the certified seeds. So it's very much important that the maintaining the nucleus seeds and breeder seeds genetically pure. So depends on the, when we talk about the seed production in agriculture, there are different types of crops depending upon the modes of pollinations. Depending upon the modes of pollination, crops may be of self-pollinated and cross-pollinated or even ovid cross-pollinated. So we deal with the maintenance of self-pollinated crops as well as the maintenance of cross-pollinated crops separately. So while we talk about the maintenance of nucleus and breeder seed production in self-pollinated crop, we know very well that the self-pollinated crop is considered as the completely homozygous and homogeneous. These two words, students, must be known to us, that homozygous and homogeneous. Homozygous means the genes are at homozygous level, homozygous at each locus, whereas in heterogeneous conditions there must be uniform populations. The variations, even after, the variations occur due to several reasons, that mutation, that's natural crossing, that mechanical mixtures, etc. And particularly the genetic mixture is maybe due to the newly released variety. The process of newly developing the newly released variety, if a genes are heterozygous at a particular locus, may segregate in the next generations, so that will create the variations. So it must be essentially purified. So the maintenance of nucleus seeds of newly released variety, the Harrington suggested the procedures in the year 1952. There are different steps in maintaining the nucleus seeds of newly released variety. We should just underline that newly released variety which are just released, but their seed production comes under the seed production chain. So there are different steps. The first step is the sampling of variety to obtain nucleus seeds. Sampling means that we should take some of the varieties for which the seed will be purified for as nucleus seeds. So for this sampling of promising variety is done, we take at least some of the promising variety into production process, so that they take the maximum of 50 varieties will be taken in one crop seasons at one location, because until unless we take a large number of varieties for this nucleus purifications, it's very much difficult to maintain that. That's why we should take 15 varieties, newly released varieties, for this purpose. And we know that each and every variety should have a promising, have potential in the production. That's why it must be promising for that particular regions. And for that, we conduct a replicated yield trial. For examples, we take the 15 varieties, it will be conducted in a yield trial. replicative elytra, suppose this one and this one, likewise this one. These all are the R1, R2, R3. This is the experimental principles of experimental design, that when we conduct the experiment for elytra, it must be replicated so that each and every genotypes perform unbiased. So we just plant, we grow the 4-5 rows of a single variety, and the difference between the distance we maintain. Some of the varieties, different varieties, we should maintain like this. So likewise we grow this 4, 5, 15 varieties. So likewise in this randomized application is that only the random allocation of the entries in a particular block, or replication is the repeated allotment of this genotypes. So we conduct the IVth trial, replicated IVth trial, and simultaneously at maturity, when the crops are going to mature, we harvested 3 meter row, we harvested border rows, we harvested 3 meter plants, we harvested the plants which are at border rows like this 3 meter central border rows. We just harvested. Suppose this row is of 5 meter length, then we just consider, we take in account the 3 meter central rows of border rows. Border rows means that the lines, the rows which are outside the outermost part of this plot, because this distance is more, and this particular plants present in this particular row have more soil, more space, more air, more temperature, etc. That's why it is not considered in the IVth. We will take only the 2 rows yield of in IVth, 2 rows for IVth. So we harvested 3 central rows from the border rows of each and every replications. So it will continue. Then second, we just take sample this 200 plants, minimum of 200 plants from this, and those plants which have less number of dealers, which are diseased, that must be discarded. And thereafter we just consider, we just take these 200 plants, and thereafter it will just harvested at the time of physiological maturity, and then each and every plant, sample plants are separately kept, separately wrapped in a cloth and make a bundles. We keep separately, and then simultaneously we have the IVth trial data, and which one are good, which one out of 15 varieties, which variety has much more yield, we just consider at those varieties. Suppose we take 15 varieties here in IVth trial, so after that we just discard 5 to 6 varieties and take 9 to 10 varieties into next generations for the potency purpose. So this way we just kept these all the bundles of all those potential varieties separately, and thereafter these bundles are also again traced separately. After tracing and considering the potential varieties bundle, we go for the next step, that is the table examinations. Table examination means the harvested, the threshed material of each and every plants are being examined properly on a table. On a table, we make a pile on this table. The seeds are arranged in this way of each and every plants. The seeds are being arranged in this way, make a pile, and then we critically examine this pile. If there is any admixture of color or any texture is present in this pile, we just discard the whole pile like this. We just discard and select only those piles which are free from any contamination or any off colors. So the table examination is very much essential, and then we after table examination the piles or the plants which are good and true to the type would be just carried forward for the next step, that is the locating and seeding of nucleus. We make a nucleus lesson of these 200 plants. Out of that, if we just discard some of the plants, suppose we discard 20 plants, then 180 plants we have, or suppose we discard 50 plants, then we have 150 plants. These plants are sown in a seedling nucleus lesson, and for that there are some of the criteria in the selection of plants. That first is the preceding crop requirement. We earlier told in the seed production principles that the principles of preceding crop requirement is very much essential, because if we have a crop, the same crop in the particular plot, and in the same seasons next season we are just continuing the same crop, growing out same crop, there will be the chances of contamination. So preceding crop requirement is must. We just check in the lands. We select the land, those lands which are free from any weed seeds, and previously in the same crop was not grown in that particular plot. Second, that the land must be fertile. We know very well that the fertility plays a greater role, very good role, vital role in the production purpose. That must be fertile, clean, free from any debris, and constantly teach all things. Then all those selected bundles or plants will just grown in a 4 blocks. We just make 4 blocks like this, and those selected plants are grown in a double row plots. So the seeds of single plant will be grown in a double row plot, and then like this. The distance between these two plots should be 45 centimeters. Must be a space. The purpose of this space is that we critically observe each and every plants from these double row plots. Likewise, so the distance between the two plots, must be enough distance, so the movement of persons which are related with this purification should be free. Purpose is only to observe critically each and every plants for their plant height, for their flowering, or other purposes for which the variety has been developed. So this way we grow the double row plot in 4 blocks, and then proper isolation is applied. Very nice. It must be isolated from one crop to the other crops. It means the same species. We must know about the species should be should not be same. So proper isolation is done. Then next step is inspection of seed plot. Each and every plots are inspected very nicely, and then critical observations for plant height and all these things. Disease reactions should also be considered, taken into account during this field visit, during the field inspections. Then harvesting and threshing of nucleus seeds. The remaining plants harvested separately. All these are, if out of these some of the plots showed the differentiation, variation, then we discard these plants, whole plants. We discard like this, and the selected one, the plants which showed the uniformity to the types, would be just selected and make a bundle and harvested properly. The harvesting time would be at the physiological level of harvesting, 5 days prior to that of the full maturity. So bundles will be nucleus level and stored. Rejection of bundles to poor yielding varieties, there should be there. Individual plant threshing and subjected to table examination. Similarly, we again go for the table examination, which I earlier told that we make a pile of each and every the seeds of each and every plants, and then table examination will be carried out, and then again we select, we discard those some of the less yielding variety. The second year simultaneously the yield trial will be conducted. So on the basis of 2-year data, we will confirm about the potential of a particular variety, so that the less potential variety would be discarded, and the rest one will be bulked to have a breeder seed stock, which are at the hand of breeders for the production of breeder seeds. So maintenance of, now next is the maintenance of breeder seeds of newly released varieties. Once we have the breeder seed stocks, we can multiply this in breeder seed of these newly varieties. So the breeder seed stock, the seeds from this breeder seed stock will be shown, and the seed sources will be this nucleus. Clean field, as I told earlier, that every time you must consider the preceding crop requirement. It must be free from, the plants must be free from any disease and pests, and it must be clean, fertile, and the previous crop requirement should also be considered, what I told earlier, and I am telling that best farm practice, whatever the agronomic practice, scientific agronomic practice to get a higher yield, it must be taken care of, and for this we provide the different agronomic principles, what I told in earlier lecture, that there are different agronomic principles, that the irrigation, the fertilization, the weed, and so a number of the agronomic principles are there. We should follow the proper best farming practices. Isolation is also very much essential everywhere, every time. These are the technical know-how being practiced in the seed production. That isolation, roguing, removal of types. Isolation, you know very well that there are 2 types of isolations, that the time isolation and the distance isolation we don't have. If we don't have a larger space, larger area for the production of number of variety at the same time, same year, same location, then we go for the time isolation, just deferring the flowering times. So removal of types is must before the flowering, must harvesting at proper times. We told earlier that the harvesting must be done at physiological maturity and before 5 days of full maturity. These will be threshed properly, and cleaning the processing, seed processing, it is one of the important process of seed production, that the processing part. The seeds are processed and clean and bagged, and this leveled with the proper tag. We know very well that the breeder seed is tagged with the golden yellow colored tags. It must be known, must be underlined that the breeder seeds are labeled with golden yellow tag. Then purification of breeder seed stocks. Suppose we have required the advancement, the more quantity of breeder seeds in the next few generations, then we can go 1 year more for the multiplication of breeder seed stock to that of the breeder seed stock. But it should simultaneously be purified properly, and for that we select some of the 200 plants similarly, and then they are grown year to row methods, and then any variation occurred in the particular lines, particular rows, that would be discarded. This way we can purify the breeder seed stocks. These are all about the maintenance of nucleus and breeder seeds of newly released variety. Now the next step, that if we have the established variety, that also the seeds will be required. So the purification, the genetic maintenance of these nucleus and breeder seeds is also essential of the established variety, which are being performed good at a commercial level at farmers' fields from 2 years, 5 years, likewise. So these are also maintained by raising a good crop in isolations and by bulk selections. We just grow the crop in isolation, and second method is the bulk selection. For bulk selections, there are different methods what we studied already in third semesters, that there are different types of bulk selections. One type of bulk selection is the compartmental bulk selection. We make a whole plot in a compartment like this, and from these every compartments we select some of the equal number of plants from all these compartments, and then these selected plants would be bulked together to have a breeder seeds stocks. So this way we can maintain the breeder seeds of established variety. Maintenance of nucleus and breeder seeds in cross-pollinated crop. These all about the self-pollinated crop. We know very well that in the self-pollinated crop, we are generally taken care of about the open pollinated varieties. But there are, for example, in case of rice, we are also using the hybrid. So in this way, the development of hybrid, the seed production of hybrid seeds are different to that of the normal process of this maintenance of nucleus and breeder seeds. So maintenance of nucleus is based on the method of development. We know very well that in case of cross-pollinated crops, the varieties are developed like hybrids, composites, and synthetics. There are three types of varieties generally we handled in case of cross-pollinated crop. The farmers are growing these types of varieties, hybrids. The hybrids are, we know very well, this is the F1 generations. This is the F1 generation, the superiority over the parents. This is the hybrids. So this way, this may be single cross hybrids. This may be double cross hybrids. We already know, already studied in third. So semester, the different crosses, different hybrids, that single cross hybrids, when we use two parents A into B, the product would be the single cross hybrids. If we consider the double cross hybrids, then A into B, when F1s of these processes we consider that C into D. Here we use 4 parents. So like in 3-way process, we cross single cross to that of the inbred lines of C, like this. So this way the top cross also there. The top cross is generally used for the testing of lines, the potentiality of particular lines. When the single cross is crossed with the testers, as testers is the open cross-pointer variety will be just considered as a third parameter. We take just drop cross methods of hybrids. So this way, double cross, single way, we use this hybrids. Okay. Next one is the composites. We know very well the development of composites and synthetics. We can synthesize. In case of composites, we cannot make the same variety, they develop the same variety in the new, in the next generation. But synthetics are synthesized again. Synthetic variety can be resynthesized because the parental material used in the synthetic varieties are estimated estimated for their combining, assessed for their combining ability, the nicking ability of a parents to nick good characters and transmit it to their progeny. So combining ability, that GCA, gender combining ability, and specific community. So based on these methods, the development, the maintenance of nucleus and breeder seed would be varied. The steps of there are several steps comes under this maintaining nucleus seeds of inbred lines. Inbred lines. That's why I told that in single cross we use two parents, and these two parents are known as inbred lines. What is inbred lines? Inbred lines are single self lines, single self fertilized lines which are used, which are completely homogeneous as well as the homogeneous. It is considered as the inbred lines. So the maintenance of nucleus seeds of, first we should maintain the nucleus seeds of inbred lines. And how we can maintain it? There are different methods. The hand pollination. We use the artificial methods of hand pollination. Suppose there are different types of hand pollination. We can go for the self pollination and sieve pollination. These are two types of pollination. The result of these two are little bit same. This is differed some way, that in self-pollinated crops, suppose for example, if it is a plant and in particular suppose it is a maize plant and the female parts are here and this is the male parts. In case of self-pollination, the pollen of same plants is pollinated to that of the male parts of this same plants. This is considered as the self-pollinated crops, self-pollination. But in case of self-pollination, there is little bit difference, that if these are some of the plants, so the male parts here at the top is considered as the tassel, known as the tassel, and the female parts are at just side. So, in case of sieve pollination, we just collect the pollen from these all plants of the same variety. These are all A variety. Varieties are the same. We collect the pollen from each and every plant and mix together and then pollinate that of the female parts, so that there should not be any genetic drift. The purpose of sieve pollination is to avoid the genetic drift. Unless in case of self-pollination, if we continuously self-pollinate a cross-pollinated crop, there would be the inbreeding depressions. So it is recommended that after each and every 2-3 sieve pollination, we once go for the self-pollinations. These are recommended in the hand pollinations. So after hand pollination, the plants which are very much true to the type, the crops will be harvested. Table examines each and every crops, and the hand pollinated crops and plants would be harvested, and then table examination is done. Like I told earlier, that each and every crops, the seeds of each and every crop. First we arrange the seeds, arrange the crops in a pile. Suppose these are the crops. We arrange this self or seed or seed-pollinated crops in this way, and these are table examined critically for the off color and off texture. It's very much essential to see the differences, the variation. If in this pile, if any plants are just any seeds are of off colors or off texture, we know very well that there is the Genia effect. There are different textures in case of maize. So if the seeds are different, it differ from the original one, it must be discarded. The whole pile would be discarded. So after this table examinations, the seeds are again then seeding going for the nucleus nursery. It is known for it is required to be raised a nucleus nursery like the same. We must have the requirement, the critical, we should must consider the critical requirement of this nucleus nursery, that preceding crop requirement, isolation, roguing is also much very essential. We should always visit the fields to take observations on any differences, any variations present before flowering. There must be twice visit, 2 to 3 times visit, we can visit, and then harvesting is done. We harvest the plants year to methods of these, the selected self-pollinated crop, the hand-pollinated crops would be grown in a year to methods. The single crops, the seeds of single crops is grown in a single row like this, and proper field inspection is done. And after maturity, at the time of after maturity, when we are going to harvest this single rows, the materials are just arranged here in this way. Each and every, the closely of each and every rows are just arranged in this way and critically examined. So if there is any variations present in this, we can discard all this. The closely of all these could be discarded and select this one, this one, this one, like this. So this way we just maintain the genetic purity, and its critical examination for colors and texture is very much essential. If there is any differences in colors or any textures, it is supposed to go for the self-pollination again. Thereafter, the bulking of seeds, bulking of these. Suppose this is good, this is true to the type, we can bulk the seeds of this like this. We can bulk the seeds of these lines. So these are clean, treated and stored in ideal storage conditions. These are very much important. The nucleus seeds, it's very much useful, costly affairs. So we just store the thresh properly and then clean this storage stored properly. The maintenance seeds of non-inbred plants. As we told earlier that depending upon the methods of development of variety in cross-pollinated crops, there will be the development of the hybrids, the composites and the synthetics. So in case of non-inbred plants, we just make a plot, find the we have made a row plot and this, and similarly this we will go for compartmental, and 5,000 plants is selected. 5,000 plants would be selected from this area, and for this its small plants are seed planted, and this we select some of the equal number of plants from each and every compartments, and then the selected plants from each and every compartments are shift pollination. It means we collect the pollen of different pollen from different plants and pollinate the selected plants. So we pollinate these selected plants, 5,000 plants, and this we should always be taken care of about the isolation, about the preceding crop requirement, because these two are very much essential to avoid any contamination, any impurities at genetic levels. Okay, students, it's a must be considered, taken care of, always taken care of in your mind, that the isolation and preceding crop requirements. 5,000 plants are shipped. The roguing is also being carried out during this crop seasons, and persons which are well known in that of the materials will be visited in the plots, so that they can identify any off types. Careful examination for off colors, textures and diseases. At the time of this crop seasons, we can also take care of about the disease incidence. If there is any diseased plant, we just remove that, and after harvesting, we just taken care of about the off color and off textures, and desirable ears which are selected from this 5,000 out of these 5,000 plants. The desirable pods, the desirable ears are dull and dried and shelled. These are then cleaning is done, and the seed treatment and storage is done for this. Next one is the maintenance of breeders' seeds of inbred plants. Breeder seed stocks obtained from the nucleus seed plants is again multiplied in the breeder seeds stocks. So that are the same, the land requirements, the isolation, roguing, and all the agronomic practices to raise a good crop would be done, and avoid any contamination. Harvesting, soil, and sorting, drying, and pressing is done in case of maintenance of breeder seeds of inbred plants. Maintenance of seed of established variety in case of cross-breeded crop. Again, as we know that there are established variety, there is the composite and synthetics of or hybrids even are established one, not newly released. In that way, we can again raise a breeder seeds in isolations, and then again mass selections and modified plant selections is being implemented for this maintaining breeder seeds of established variety. So this way we just finish about the maintenance of nucleus and breeder seeds of different set quality crops and breeder seeds. Thank you very much.